welcome to BIOS Roundtable. We are so excited to welcome back two great friends of the show, in Dave Schaffer and Samir Mitragotri, both world-renowned scientists, esteemed professors, and serial entrepreneurs. Thank you both once again for joining us. It's an absolute pleasure to host you.
Thank you.
Pleasure to join.
To help host this episode, I'm joined by my colleague, Drew Sharp. Let's kick things off. Dave, can you share a brief intro with us?
Sure. I've been a professor at Cal now for 24 years in bioengineering, molecular and cell biology, and chemical engineering. After training at Stanford, MIT, where we overlapped, and then at the Salk Institute for a postdoc. So our work focuses on gene and cell therapy, and in particular, a lot of work in gene therapy. And about 10 years ago, we felt like that work was potentially ripe for translation into the clinic, which to me meant that we needed to start companies, because companies are the entities that take technologies from academia, get them into the clinic, and then hopefully eventually get them FDA approved. So that started me on my path of founding companies. So I've co-founded 8 so far, and technologies that we've developed are now being used in 9 human clinical trials.
It's a phenomenal sentiment and something I think we can all relate to and are excited to dive into. Samir, would you mind sharing an intro of yourself?
Sure. So Samir Mitragotri, by training, I'm a chemical engineer, got my undergraduate in chemical engineering, came to MIT to do my PhD. That was my first entry into drug delivery. Did my PhD with Bob Langer, worked on a technology to deliver drugs transdermally, and was very excited to see this technology go to the clinic. So we started a company. That was my first startup. I actually worked in the company for a couple of years. It was very educational to see how technologies go from the lab into the clinic. After a couple of years, the technology advanced. It eventually got approved for a clinical product. But I was always motivated to go back to academia and be in that process of innovation and translation. So moved to UC Santa Barbara in chemical engineering, where I was a faculty for 17 years, and moved to Harvard and Wyss about 6 years ago. Currently, I'm a professor of bioengineering at Harvard School of Engineering and the Wyss Institute. We work on drug delivery technologies of different kinds, from small molecules all the way to cell therapies, and have been very interested in kind of coming up with new technologies that can solve important problems in therapeutics and figure out a way to translate the technology into the clinic. So have started a number of companies over the years, many of which have products out there in the market as well as in clinical trials. It's a pleasure to be here.
And a pleasure to have you. I'll now pass it off to my co-host, Drew, to guide us through our first topic. Drew?
Thank you so much, Chris. Dave and Samir, thank you again so much for joining us. We're so excited to host a fantastic roundtable with the both of you. We've covered much of your storied past and the significant impact generated through your work in our podcast earlier. I highly recommend our audience take a listen. But to really continue the conversation and catalyze this further, we wanted to approach your research through another lens. I mean, really through this roundtable series, we wanted to hold the discussion and debate to really have a rounded discussion around the future of genetic and cellular engineering. But really, before we dive in here, I would love to set some context for our audience. Just to begin here in ground set, how has the field of genetic and cellular engineering really evolved over time from your own lenses, from your own perspectives? Maybe just to kick things off, Samir, would you want to start?
Yeah, I mean, I think it has come a long way. I mean, I have been involved in science for almost 30 years. Actually, that's when I first met Dave, when we were graduate students at MIT. And when we were graduate students, there were no gene therapies, there were no cell therapies. And from that time, now we look at the landscape, I think there are maybe over 2,000 clinical trials in cell therapies, many in T cells, many in stem cells. There are over a dozen products in gene therapies, and I think 300 clinical trials, many clinical trials in RNA therapeutics, over 200 clinical trials, notable products out there. So that's a tremendous progress. A lot of that has happened in a very short period of time, going back maybe 5-10 years. So it's incredibly exciting to see this rapid progress. And maybe I would say, some of the therapeutics that probably were not even conceivable, when we were sort of starting doing science, to see them in reality and many more in the pipeline. That I would say is remarkably strong.
The progress has been absolutely astronomical. And just from your own perspective, Dave, things to add.
Yeah, so I mean the products in this space, of course, have two parts to them. There's the the content, the cargo, and then the delivery. And there has been pretty remarkable progress in both of those over the past 30 years of our lifetimes. And if you think about the cargoes, RNAi, was discovered roughly around the turn of the millennium, around 2000 or so. Received a Nobel Prize not long thereafter. First RNAi drug that was approved 2018, and Alnylam now has 5 approved therapeutics based on RNAi. Gene therapy, this dates back in a sense even further, where people like Francis Collins were cloning genes whose mutations resulted in genetic disease, cystic fibrosis, as well as Huntington's were cloned by Collins. And then the concept is, can you drug this knowledge, turn it into a cargo that can begin to treat a disease? And we have, we've had a number of of clinical trials for cystic fibrosis, not a cure yet, but there has been progress. And then, recently, of course, there's the initial development of genome editing tools, and then the kind of democratization of that technology with the discovery of Cas systems. And now, this year we're going to have an FDA approval of a genome edit for the first time, which is going to be for sickle cell from Vertex and CRISPR Therapeutics. So that is remarkable progress. I would say, and I think Samir would probably agree wholeheartedly, given that both of us work in this area, a lot of these advances were unlocked by the second part of the technology, which is delivery. And each one of these advances was enabled by a delivery tool without which it would not have succeeded. And in many situations, there's this old joke in the gene therapy field, there are only 3 problems: delivery, delivery, and delivery. In many situations, the cargo comes first, and then is the bottleneck or the rate limitation becomes, how do you get it there? And I think we're at that point. We've always been at that point for the past 30 years, but we're most definitely at that point more than ever. So I think that looking ahead, we have our hands full to continuously improve the delivery and fully unlock the potential of these technologies.
David, those were some fantastic applications that you mentioned within genetic and cellular engineering. I really appreciated kind of the overview and the quick evolution. But to ask you directly, I'd be curious, from your perspective and background, could you maybe dive through maybe just some of the greatest applications thus far that have been enabled by genetic and cellular engineering from your perspective?
Yeah, I mean, as Samir mentioned, we have, roughly a dozen FDA-approved gene therapies and, multiple RNAi therapeutics that have been approved. And, I think that what the field has learned is that you don't necessarily always go for the disease with the largest patient population. It's tempting to do that because there's so much unmet need, but it's good to go for the quickest route to be able to show a technology can potentially work, the quickest path to de-risk the technology. And that in some situations is not always going to be a very large, highly prevalent disease. So we work, just to provide a specific example, we work with AAV gene delivery a lot. And the first FDA approval for an AAV gene therapy was for Leber's congenital amaurosis type 2, and they were able to extend the label as well to retinitis pigmentosa. But initially, LCA2 is quite literally a 1 in a million disease. The prevalence is 1 out of 1 million births in the United States. So that's obviously not going to be a huge product for Roche, which eventually acquired the company that developed this therapeutic, but it shows it can be done. And there was a quicker regulatory path, really well-developed animal models, a highly networked patient population, clear guidance from the FDA on approvable endpoints. And so all that combined together, I think, to result in an approval that showed this can be done, you can do this. And that really, I think, catalyzed and galvanized the field. And so we've now, since that time, had 2 more AAV FDA approvals. There are more in the pipeline. And I think that's the case as well for these other branches of genetic medicines, where you don't necessarily always go for, cancer or heart disease right off the bat, because those are really challenging. But maybe starting out with an orphan indication where there's a clear path to approval, and then building up momentum from there, and going after tougher targets that are going to progressively have more and more of an impact on medicine.
And to turn the table to Samir real quick, we would love to ask you specifically, what are the current challenges we're facing today in genetic and cellular engineering? Could you give our audience some context?
Sure. There are many. You can identify challenges from identification of the right target to manufacturing. But I think one that really comes to mind, and this may be because of my own bias in delivery, and I'll build on to what Dave said, is targeting. And the reason I think it's really important is that if you look at how drugs have changed over the last 25, 30 years, there used to be small molecules, then they became biologics, and now we are talking about genes and cells. And that really brings upon a different challenge for delivery and targeting. A small molecule hangs around in the body for a short period of time. If you deliver it somewhere else, it's a problem, but maybe a short-term problem. If you deliver a gene that can be permanently inserted into the body at the wrong place, you're going to have to live it forever. So that makes it extremely important that we hold the bar to targeting very high. And for cells, I would say that bar is perhaps even higher. And if we look at the delivery efficiencies or targeting efficiencies of current strategies, they are low. They are potentially workable in some diseases, and that's why we see products out there. But there's a tremendous need to make them extremely efficient and specific. And that I see as a big challenge for biologists and engineers to work together and solve in the future.
100%. And just focusing on applications to begin with here, as you have mentioned, from the impact and the current applications, the field itself is broad. But really the applications from novel therapies specifically derived from genetic and cellular engineering, even in just the last decade, have been the engine behind many transformative companies currently impacting patients in the clinic. It's fantastic to see the progress. Just a quick question to dive in here, and maybe Dave, you could start. Maybe you could just explain to our audience, or just start here, why are cell and gene therapy as a field itself different from other modalities?
I think it relates to what Samir just mentioned. This is both the power of the field as well as the risk. That, like Samir mentioned, small molecules have a short pharmacokinetic half-life. They last a few hours. And as a result, they're not really a cure, right? You have to pop a pill every day the rest of your life if you have hypertension, for example. As you move on to biologics like antibodies, you've extended the pharmacokinetic half-life from hours to days or perhaps a couple of weeks. And that means your therapeutic has a longer effect, but it's going to go away eventually. Again, it's a treatment, it's not a cure. And so what is, transformative about gene therapies and cell therapies is that the therapeutic modality, the drug itself is DNA in the form of a gene or an entire genome. And DNA, we know, unlike proteins and small molecules and even RNA, has the potential to stick around for the life of a patient. So the power, of course, is that you can begin to think about cures, especially in situations where you know enough about the underlying mechanism of the disease that you've designed your molecule right to treat it at the root cause of the disease, and where the delivery is good enough that you can deliver it to the point where you've transduced or infected enough cells to modify underlying disease progression. So that's the power of it. You can begin to imagine a world where it's one and done, single treatment, long-term therapeutic benefit. And of course, that's the risk as well, that it's hard to undo that. The field is, working on ways to get far better targeting, as well as to have, quote unquote, kill switches to enable the cargo to go away if you'd like it to. But right now it's one and done, mostly for better, on occasion for worse.
Really well said, David. Samir, anything to add to that perspective?
Yeah, maybe one quick point. I think, when it comes to cells, I think the uniqueness there is that cells have their own agenda. I think for the first time in the therapeutics landscape, we are dealing with drugs which have their own agenda. I mean, small molecules can degrade, proteins can unfold, but that's about it. The cell can become something else. And that really poses a question as to how do we control the cell? If you think about the delivery strategies of the past, small molecules had their own challenges, biologics had their own challenges. But when it comes to cells, we have to kind of think about a different level of challenge. And that really makes it very exciting for the delivery sort of field because it poses new challenges. But at the same time, I think those challenges will have to be met for cell therapies to really deliver their full potential.
100% agree. And it's amazing to see the current challenges that we're facing within the field itself to really unlock the full potential. Just as a continuation of some of these issues that we're bumping into and why cell and gene therapy and genetic and cellular engineering is so unique as a modality itself. I'd be curious to follow up, Samir. How does this need for specialization within the modality itself really change your scientific approach when you're dealing with developing these new and novel therapies?
So, the field is evolving in multiple ways, and everybody in the field has their own unique view and contribution and the technology to really help solve the problem. In our case, a lot of what we had done in the past is based on polymeric systems. I have been working in sort of polymeric nanoparticles to deliver drugs. And we sort of started leveraging that in the context of cell therapies. And the question that came to our mind is that, when you grow cells in vitro, in whatever your favorite culture system is, let's say 96 well plate, there is a tremendous control on what you do to the cells. You can provide them with the support you want. You can provide them with the nutrients you want. But when you inject them in the body, you don't have that level of control. So that really posed the question in our mind that, ex vivo, it's still a polymeric system. Can we leverage that? So with that in mind, we started developing systems that we call backpacks. These are basically polymeric system that can attach to cells and can control the cell behavior in vivo. And in many ways, if you look at the core material, it's still the same, right? It's a PLGA or polymer, but the way to use it and the way to kind of use it in the context of cells, I think has been changed because the drugs have gone from small molecules to cells. And there are probably many other examples of researchers around who are kind of using the tools that we as a drug delivery field have learned over the last 25 years and looking to solve the problems which are here today and kind of represent the problems of tomorrow.
Yeah, and as you mentioned, Samir, every lab has a different focus, a different approach. Maybe if I could turn that to Dave real quick, would love to hear your own perspective and potentially how you amongst the Schaffer lab are addressing these challenges in your lab today.
I think one theme that has emerged and that we're certainly strong proponents of is that these are complicated problems, right? Extremely complicated. And if you consider delivery as a problem, consider an antibody by comparison. The job of an antibody is to circulate around, typically in the bloodstream, sometimes in other regions, such as the eye, and to bind to something and to inhibit it. And so it has to have one part of it that controls circulatory half-life and another piece of it that controls, specificity of binding. And the circulatory half-life is typically controlled by the Fc domain, which antibodies kind of gave us for free. That's how they're designed to begin with. Consider a delivery technology, a nucleic acid delivery, and let's say you're trying to go IV to the heart. Well, you have to, first of all, manufacture it. It's a lot more complicated than an antibody. It has part nucleic acid, part protein. It's the processes to manufacture it are inherently more challenging because it's difficult to create a viral particle. You have to inject it. It has to biodistribute to the right places, typically avoid the liver if you want to go to the heart. Then extravasate, diffuse through the tissue, find the surface of a target cell, ideally have some level of specificity, which is really tough and not currently that feasible. for, let's say, a cardiomyocyte, then get internalized, escape the endosome, traffic to the nucleus, pop out the DNA, and persist for a long period of time with a drug product that is a complex promoter driving expression of an RNA or a therapeutic protein. So that is way complicated compared to an antibody, which is, why the field has taken time to develop. And, to be fair, it took time to develop monoclonal antibodies in their day as well. So really, if you're faced with that complexity, one solution to try to address it is to study the problem at a very detailed level and try to hypothesize certain steps where you can intervene or engineer or improve. And the other possibility is to approach it the same way nature did, because, after all, nature did tons and tons of protein engineering over 4 billion years in the absence of a protein crystal structure, and to do throughput. So throw many potential solutions at the problem and see what works, and use iterative diversification and selection of large pools until you end up finding a winner. So we started doing that with gene therapy back in 1999. I think analogous approaches by colleagues in the field have, brought throughput to bear on non-viral delivery, on liposomes or lipid nanoparticles, as well as polymer nanoparticles. And, in other branches of the field, the complex task of taking, let's say, a cell and turning it into a differentiated specialized cell, that is, people are applying high throughput to that because it's a very complicated problem to understand. So for a variety of reasons, there have been highly enabling technologies that have enabled us to apply throughput to these problems and to start finding optimal solutions to them. And I certainly hope that continues, empowered by, advances such as sequencing and machine learning in the future.
And excited to dive into the future later on. I think could not have asked for a better ground set to kick off this discussion. Thank you once again. Going to pass it off to Chris to dive into the specifics here on our second topic. Chris.
Thank you, Drew. Thank you both. And as we dive in a little bit further and talk about, just as you were just saying, Dave, a little bit more in the delivery space, taking a step back, you both share the same North Star of therapeutic impact, but you've tackled this problem in a relatively different manner, as we've already started to discuss. Dave, with much of your work coming from AAV viral delivery and stem cells, and Samir, your work consisting more in the small molecule and novel cellular approaches spaces. And so as we think about this shared North Star, and we'd love to use this opportunity on the roundtable as a way to come together and discuss some of the similarities, differences, and applications of both viral and non-viral delivery. So I guess to begin with, and maybe Samir, if you'd like to kick things off, we can start. Where are we today in terms of delivery?
So I think, we are at a very good place, Chris. So, when we talk about viral and non-viral vectors, there is always that sense of competition between the two. And I think that is good because when science competes, patients win, right? It really drives it raises the game. So if you look around, there are plenty of examples of success stories with both viral and non-viral vectors. Viral vectors, Dave is a pioneer, so he'll speak more to that. Non-viral vectors, we have seen the success of mRNA vaccines over the last few years. So overall, I think there has been tremendous progress made, both with respect to the design of these vectors and what they can do. Clearly, there are going to be differences between the two. There are going to be inherent strengths and there are going to be inherent limitations. But overall, I think both these technologies, viral and non-viral vectors, have shown the promise and delivered on the promise that they're capable of. So I think I see that's really a sort of a great victory for these technologies.
I think that's a great overview, Samir, and would love to turn it over to Dave to ask a similar question. Where do you think we are today in terms of delivery?
Sure. Yeah, I think the way I view these in some ways is not competing. I actually worked on non-viral for my PhD thesis, worked on retrovirus, lentivirus, AAV, adenovirus, vaccinia. And some of these, we're no longer pursuing because they weren't the answer to the challenge. But I kind of view delivery as Samir does, which is it's an enormous field, right? There are many molecules that need to be delivered, and there are many contexts in which we need that delivery. Sometimes we need it to be transient, sometimes we need it to be permanent. And so for any given problem, we should pick up the right tool and use it. So in the case of AAV, if you want to deliver something that's under roughly 5 kb to a non-dividing cell and have it last the rest of somebody's life, AAV is your vehicle. It's increasingly becoming so as we create new versions of AAV that are targeted to a variety of tissues in the body. If you would like to deliver a genome editing tool that needs to be present for a few hours to introduce a precise edit into the genome, and then you want it to go away so that it doesn't start doing off-target, doesn't start causing immune responses, because Cases, after all, are bacterial proteins, then you probably shouldn't be using AAV. And non-viral is looking pretty good. If you want to deliver a vaccine and you need a booster 4 or 5 times, as we've seen recently, viral is probably not the way to go, because our immune systems at the same time as raising immune response against the cargo of the vaccine, spike protein for coronavirus, we're going to raise antibodies against the coat protein of the virus. So the virus is going to be neutralized the next time around. And that's, non-viral is the best way to go there. Non-viral is having challenges currently achieving long-term genetic introduction into cells. That's always been a challenge, and efficiency is a challenge as well. But I think that as the field continues to progress, both non-virally as well as virally, I'm going to view these as, what's the best option and what's the best opportunity for the given, genetic engineering task you're trying to carry out.
I think that's well said from you both, and that for each intervention, there's an ideal toolkit, and hopefully we can help bring those parts of the toolkit forward to really benefit patients most of all. And so as these technologies progress, and David, you were just talking about things like around some of the challenges around size of payload, specificity, immunogenicity, and as we start thinking about the progress of these delivery technologies, how do you think they scale, especially their delivery mechanisms? How do you think they scale over time?
When you scale, do you mean scale to patient size, or are you talking about genome size, patient numbers, or genome size?
I think that's a great distinction. And I was thinking from the lens of biomanufacturing, especially GMP scale up, so they're able to reach patients. But by the same token, I think it's a great question to talk about when we're saying payload size as well. So why not tackle both perspectives?
So, I'll talk a little bit about viral and love to hear Samir's perspectives on non-viral. Non-viral, you're in the world of chemical synthesis or, biochemical synthesis in the case of messenger RNAs. So it's going to be cheaper. In the case, so that means that you need to pick your battles with viruses correctly so that it's well suited to the therapeutic target that you're trying to address. So again, AAV, the goal is to get permanent genetic modification of a non-dividing cell with under 5 kb. And right now, it's a very expensive therapeutic, right? We just had Hemgenix from Unicure. I was on their board of directors for 6 years. Stepped off in 2020, but Hemgenix was just approved, $3 million. And that is, initially one gets a little bit of sticker shock looking at that. At the same time, the standard of care for hemophilia B is a recombinant protein that costs $250,000 per year. And fortunately, due to that recombinant protein, hemophilia patients are living a normal lifespan. So you take 75 years, multiply it by $250,000, we're talking $20 million over a lifetime, not even counting for inflation of drug-related cost. So $3 million is not that, bad. It could be viewed as saving the healthcare system resources. So that works well for hemophilia B. And in general, as we know, drug prices for rare disease, which is where gene therapy is by and large started, tend to be higher because the system and the Orphan Drug Act want to incentivize companies to develop therapeutics for otherwise neglected patient populations. So the market will tolerate a higher price, so that higher price can absorb the high cost of AAV manufacturing. it's really my hope that, we're going to gain momentum helping patients with rare disease, but really to unlock the full potential, we need to move on to diseases that affect larger numbers of patients. And if this is the case, the cost of manufacturing, the COGS, are going to have to come down substantially. And there are a couple of ways to do that. One is to create a much better delivery vehicle that's targeted. So more of your drug arrives on target, which means your dosages get lower. And the second is to improve the biomanufacturing. There's a slide I have which shows the cost per milligram of monoclonal antibody as a function of time since 1980, and it's come down pretty exponentially. We're kind of right around maybe 1983 right now in the gene therapy biomanufacturing world. There's a lot of potential to do engineering of the virus, engineering of the cell line, bioreactor improvement, culture media improvement, all these bread and butter technologies the chemical engineers developed to improve antibody manufacturing. So, in the next 10, 20 years, we're going to need to bring those to bear to improve, the manufacturing in the next generation of biologics to make them much more accessible to large patient populations.
Yeah, and to make a quick comment on the non-viral side, I mean, we all got to witness the scalability of the mRNA vaccines, right? I mean, tremendous scalability in a very short period of time. But I think we have to keep in mind that didn't happen overnight. To build on the point that they made, that the cost comes down as we learn more. And in the world of lipid nanoparticles, the history goes back a long time ago. So there were cationic surfactants that the industry was aware how to make at scale. Then came cationic lipids. They were first used for siRNA. And by the time we got to the mRNA LNPs, there was tremendous expertise in making them, scaling them, and characterizing them. So all that expertise helped in scale it, both with respect to the number of patients affected, but as well as cost. And that's the game, that the more we familiarize ourselves, invest in understanding technologies, cost will come down. And, that's sort of, it has materialized in pretty much all other forms of engineering. And I don't see any reason why it wouldn't do so for any delivery technology that we are thinking about today.
Keeping in that same vein of delivery, but pivoting slightly, we'd love to get your thoughts on a few areas for quick rapid fire. And so would you say the following are overrated or underrated, and why? Extracellular vesicles and exosomes. Samir, do you want to start?
Wow, you want to put us on the spot, don't you? It's a tough question, but I think extracellular vesicles are interesting. They certainly have their own capabilities, but I must say I do get kind of concerned when people have unreasonable expectations from new technology. EVs are lipid vesicles, to put simply. The field has had history with liposomes, so we know what they can do and cannot do. So it is reasonable to expect that EVs can do something more than liposomes have, but to think that they can solve all the problems that liposomes could not is probably potentially an overestimation.
Yeah, I would agree with that. I think raises the question, what is an exosome? And at the beginning of the field, it was, you take the cell culture supernatant and spin it down at high Gs, and those are exosomes. And we know that's not the case. You have a lot of material in there that pellets that's not an exosome. So the sophistication of the field is advanced on the biology side, so that we now have markers, we know the densities of some of these components, and there's a lot of advance in the biology. But if it's that new as a field, is it ready for clinical translation? I don't think so yet. And we've seen recent events with Kodiak that have kind of borne that out. And I do agree with Samir that there's some similarities between these and liposomes, and liposomes are very, by comparison, well defined and understood. So it remains to be seen whether or not the potential advantages of an exosome outweigh the challenges associated with characterizing it, producing it at scale, purifying it, and understanding its properties compared to liposomes.
Oh, well said both. Then keeping on you for a moment, Dave, how do we think about then gene circuits and more broadly logic-gated therapies?
Yeah, I'm always a proponent of being problem-driven. We have always wanted to go out into the field, identify what are the clinical problems, and then design technologies, the simplest possible technologies to address those problems. And if we need to add a little bit of complexity, we'll add a little bit of complexity. But it's of course taken for granted in people who do clinical development that really complicated things can break in a lot of different ways, and lots of things can go wrong because taking something into a human being and scaling it up is really complicated. So I've always been a proponent of Occam's razor, make it as simple as possible. And I think that within academia, that's not the objective, necessarily make it as simple as possible. We want to understand the complexity of our own gene circuits within our cells. People have been pushing the limits and understanding those principles and applying them to engineer gene circuits. That's all great. It's there have been a lot of advances that I've really enjoyed reading and in some cases teaching. But I will always filter that through the mindset of, if it's going to go into a human and into clinical development and into a company, it should be simplified as much as possible so that there are fewer points where something can fail.
I think it's an interesting idea and technology. And in my mind, it really all comes down to being able to control where the therapies go and what they can do. And in that regard, sort of going back to the targeting comment, how do you control where the cells are active? And one way to sort of think about is, make them accumulate at the specific site, but that can only get you so far. So having additional layers of control where it reduces the burden on getting at the right place and that entirely solving the problem, I think is helpful. In that regard, I think this is a very interesting approach. And having said that, I think it all comes down to being able to execute it to a point where the benefits are justified by the complexity.
I'll add as well that gene therapies or nucleic acid therapies, or specifically really gene therapies, are interesting and unprecedented in that you're afforded lots of additional mechanisms for control. For a small molecule, the medicinal chemists design it in a certain way, and you can kind of predict roughly what the biodistribution is going to be. But once it arrives on target, it's a small molecule. With a genetic cargo, there's the coding sequence for a therapeutic protein or a nucleic acid. There's also the promoter and the 3' untranslated region where you can embed like microRNA control. And so there are these additional mechanisms to gain control over when, where, and how much of a messenger RNA or guide RNA is produced. And that's exciting in that let's say if we consider Hemgenix again, that Unicure approved gene therapy for hemophilia B, which is a good product. It doesn't matter where it went because it's going to secrete a protein into the bloodstream. So it's the more the merrier. The more cells you get, the better. If you're trying to treat a condition within the central nervous system related to neuronal signaling, or a condition in the heart related to contractility, all of a sudden it really matters when, where, and how much. So I'm a big believer in affording ourselves of those additional opportunities for control within a gene, especially the promoter as well as the 3' elements. But I think you can do that short of assembling it into a complex circuit. There might be problems in the future where that's needed, but right now I think that the field is on a good path.
And speaking of that path, and it sounds like we're all in agreement that bioinnovation is only continuing to accelerate, be it through increasing biological understanding to the integration of tech-enabled biological solutions and insight generation. And so as industry pioneers, we wanted to ask, keeping them that sentiment of rapid fire going, what are some of the scientific trends that you're most excited about today?
Samir?
I guess one trend I'm very excited about is the convergence of drug discovery and delivery. And sort of what I mean by that is, when I was a grad student, a drug was a very different thing than the delivery technology. Like a drug device combination, for example, a solution that you put in some sort of device and inject it into the tissue. And when I look around the landscape today, I see that they are sort of coming closer and closer and becoming very integrated. Like antibody drug conjugate, for example. I mean, the recent progress in ADCs over the last few years have been phenomenal. And when you look at ADC, you kind of wonder which one is drug and which one is delivery agent, because it's all combined. And I see many cell therapies going in that direction, where strategies to control the cell are becoming an integral part of the therapy. So I think that convergence really makes me very excited, because historically, if you look at the field, the drug really took the spotlight in the therapeutic product, and delivery was kind of something on the side. So the drug delivery scientist in me is becoming very happy when we see that the delivery and discovery are kind of integrating. And I think it has something really to benefit the overall landscape, because the sooner they integrate, higher is the opportunity to discover better drugs and potentially make them cheaper and quicker as well. So that trend, I think, is super exciting.
Dave?
Yeah, I would share some of that, definitely. Or I'll just add one additional comment, because Samir already articulated it so well. Jennifer Doudna, whom I'm a huge admirer of, as is the world, I believe. So she, we recently invited her. I'm a director of an incubator at UC Berkeley called Bakar Labs, and Jennifer very generously and kindly gave a talk. Her time is very in demand. And she, of course, is the co-inventor of one of the most exciting cargos that we've seen, in the past couple of decades. And she very quickly brought up delivery. So CRISPR-Cas was discovered or published in 2012, 2013. And the field, of course, became, very enthusiastic about engineering a variety of different technologies for genome editing based on Cas9. And so we've seen innovations with base editing, prime editing, perhaps there are going to be targeted, transposon insertions based on Cas. But it's advanced to the point where I think the cargo can treat a number of diseases, and we're again hitting that bottleneck and limitation that it can't be delivered. And so Jennifer brought that up as one of the major challenges in the field. Delivery is a significant hurdle to still be overcome. I think that another innovation that we've seen, and this is nothing new, so pardon me for being a little cliche, is data and AI, right? We've seen a surge in sequencing technology within my career, within our lifetimes, and that is generating enormous amounts of data, and computation has progressed in parallel to the point where there are processors that can churn through those data to try to find patterns and try to find correlations, which is, of course, the essence of machine learning. And that's enabling a lot of efforts. One direction where I would love to see it go is for patient segregation, that, we're developing as a field these incredible therapeutics. In some situations, they're highly efficacious with a strong effect size and a high statistical significance. In other situations, the significance and the effect size are a little bit more modest. And if it's a cancer where there's a severe unmet need, the drug's still going to get approved. But wouldn't it be great if we had, technologies that would enable us to identify the patients who would be, in a position to most benefit from those technologies? And especially for expensive biologics, you'd really like to invest those healthcare resources in the patients that are going to be benefiting the most. And so I think that big data, and again, I'm not saying something that's not out there in the ether a lot already, but big data, machine learning, patient data, I think are going to really help out our fields by really enabling us to focus our energies on patient populations and diseases where we can really help the most.
I think, Dave, that's the perfect transition to pass it back to Drew to talk a little bit about the medicines to come and get your perspective on hopefully what that impact for patients looks like.
Thanks, Chris. Samir and Dave, we've been able to discuss really the North Star within cell and gene therapy specifically. I think we can all see and attain it as curative and accessible therapies. And that might be a long time away, but as we have you both on the line here, we wanted to discuss and maybe work backwards here to have a conversation about what are the steps and how can we start building roadmaps and milestones towards that lengthy and momentous goal. So working backwards here, if we could just start a roadmap. I mean, to begin with, on the first question, Dave, what do you believe really are the milestones to reaching this goal as far as getting curative therapies widely accessible through cell and gene therapy?
Yeah, I think that in a sense we've touched on some of them, which is great. I think the cargoes are the drugs are really increasingly becoming well developed. There's incredible innovation with genome editing. I've co-founded a genome editing company. It was acquired by Intellia last year. So there's a lot of progress to be made when it comes to these incredible cargoes. And where I'd love to see the future going is to democratize them, to make them broadly accessible, both within the United States and overseas. And if you think about it, a one and done therapy, well, taking a step back, we recall all the news over the past 3 years on the challenges associated with cold supply chain for the vaccines. And, one vaccine had to be stored at -80. The other one could be stored at -20. And, think about, we'd love to be able to make these therapeutics available throughout the United States and throughout the developed world. We'd love to make them accessible in Latin America and Africa in the developing world. And so a situation where you need to deliver a therapeutic that has to be kept cold and you need to administer it every day, every week, every month, that's a challenge in the developing world. One and done therapies, if affordable, are an incredible advance in the developing world. Hemophilia right now is still a problem in the developing world because, who can have the resources to be able to invest $250,000 per patient per year. So a one and done therapy has the potential to transform healthcare, both in the US as well as overseas. And it's incumbent on us to make it affordable. So that's where all of these innovations have to come. We need better cargoes that are more potent, coupled together with much more efficient delivery vehicles and improvements in biomanufacturing.
That's fantastic. Samir, any quick additions, thoughts on the roadmap, the milestones that we have to reach to achieve this goal?
I completely echo what Dave said. One comment I'll add, maybe on the basic science side. I think if we were to bring to reality new gene therapies and cell therapies, we need to understand how do they move around in the body and where do they go. So if you look historically at small molecules, we do have an understanding as to how small molecules move around the body by diffusion, by convection, and out of that came PK models. And they have been the foundation for accelerating drug discovery and drug development. When it comes to something like cells, we don't know how they move around. If you inject 1 million cells in the blood, where do they go? How quickly? And what are the laws that describe their transport? And I think we need to develop that. So I think there's a tremendous need to what I would call a PK equivalent of cell therapies and potentially gene therapies as well, although there is more known on that front. And that, I think, is going to be necessary to come from basic sciences to provide the framework for advancing these therapies.
Fantastic. And as we're on the topic of applications, I'd love to ask directly, Samir, what applications of cell and gene therapies have yet to be touched that, you really love to see tackled in the future?
I think I would say a lot of interesting and important targets have been attempted. So I don't think there is, well, you never know, never. But I think, it's hard to say that there are some targets that haven't been thought of. Having said that, enough progress has not been made for many of them. So I think, we in our lab are very excited about autoimmune as an area because that's an area where the combination of adaptive and innate immunity really plays a role. There has been a lot of emphasis on the contribution of adaptive immunity to autoimmune, but innate immunity really provides the framework for that inflammation and hasn't been as fully appreciated or tackled. So we are very excited about using cell therapies for that. And certainly oncology remains a key target for this, but there's already a lot of activity going on. But I do think that once the therapies make an impact on one area, that does provide the foundation to explore many others. So in that sense, any area where the impact is demonstrated, I think is an exciting step forward for the field.
Dave, any additions, any applications that you're excited to be pursued in the future?
Yeah, I think that I guess I'll say that, increasingly applications will benefit from advances in the basic sciences that both on the cargo side as well as the disease side. And I guess I'll start on the cargo side, right? Because RNAi was originally discovered when somebody overexpressed something in plant, expected the flower to turn purple and it turned white. And that led to the discovery of, RNA interference. Or, CRISPR-Cas was discovered because there were these interesting repetitive genetic sequences integrated into the genome of a bacterium. And people started thinking, what is that? And that's a basic, again, investigation that led to this incredible advance. So basic sciences are going to continue to enable new opportunities, new technologies. Samir touched on this on the disease biology side of things a little bit earlier, which is that, in the gene therapy field, for a number of reasons, we've really started on rare disease. And one of the reasons is the biology is so well known. If you're developing a gene therapy, you're in the same boat in some ways as a small molecule person, which is, what is my cargo? What's my therapeutic? And for a rare disease, by definition, the therapeutic is the broken gene, right? You sequence the genome, this person has a loss of function mutation in CFTR, I'm going to deliver CFTR. And that can extend into more complex disease to an extent. Wet age-related macular degeneration is about the most monogenic that you get with a complex disease. There's a well-defined de-risked drug target, vascular endothelial growth factor. It's currently treated with a very successful protein therapy or protein therapies. So it makes sense to develop a gene therapy for that, where where your cargo is, again, an anti-VEGF. So one of my companies has a phase 1/2 trial for wet AMD that's turning out quite well. As we start heading into congestive heart failure and Parkinson's and Alzheimer's, we're in the same boat as genetic therapy people as the rest of the field, that we need drug targets, which means that we need basic biology to continue to advance, to teach us what to do in the clinic.
That's fantastic. Thank you all so much again for answering, and just such an inspiring outlet just to have a conversation with you both. I want to pass it over to Chris to close up the episode here real quick. Thanks again.
Thank you both. And as Drew mentioned, just as we wrap up today's episode, a few quick closing questions. The first and foremost, do you have any additional thoughts you'd like to share? Anything we didn't get to that you'd like to share with the audience? Samir?
No, I think we covered a lot of ground. So I want to thank you for hosting this podcast. It's been a fantastic conversation with a pioneer and my close friend, Dave. So thanks for the opportunity. And we remain very excited about the future of cell and gene therapy. I think we are in the early stages of what the impact could be and would be. So to be at this point, looking at this from the technology perspective as to how we can solve the unmet needs and come up with new technologies is an exciting adventure, and looking forward to see what the journey brings.
Yeah, I fully agree. It's a lot has happened within our lifetimes and our careers. There have been tremendous advances that we both witnessed and tried our best to in some ways contribute to. But, another thing that has to happen to enable the advances across the field as a whole is for people to communicate them to a broad audience, to enable people to know what's going on within our field. So for that reason, I really want to thank the two of you, for taking the initiative to put together this terrific series of podcasts and programs that, hopefully increasingly having an impact on teaching people what the future may look like.
Thank you both for taking the time and for helping us in this journey of telling the story of the life sciences. And we're in full agreement. Cell and gene therapy is just in the opening innings, and we can't wait to see where pioneers such as yourself bring it next. So thank you both for an absolutely fantastic episode. We're very grateful for your time. Thank you again.
Thank you so much.